Dear student, today we will discuss about the disease malaria. The term malaria has been derived from the Italian words mala means bed and aria means air. It was believed that the disease was caused due to foul or bad air coming at night from marshy or swampy places. Now it is discovered that this disease is caused by a parasitic protozoa of the genus Plasmodium. Malaria parasite. Plasmodium vivax is commonly known as malarial parasite. It is an intracellular blood parasite, causes malaria fever in human. History of malaria parasite. Plasmodium was first discovered by Charles Leveren in 1880. He observed that the parasite in the blood corpuscles of malarial patient. His observations were later confirmed by Golgi and Shelley. Then, in 1897, Sir Ronald Ross, an Indian military surgeon, discovered the presence of oocysts of the malarial parasite in the stomach of a female Anopheles mosquito. For this, he got Nobel Prize. Life cycle of malarial parasite. Plasmodium vivax. Plasmodium completes its life cycle in two hosts. That is, one host is male and another host is female Anopheles mosquito. Man is the primary host and mosquito, Anopheles mosquito, is the secondary host. The female Anopheles mosquito transmits the parasites from one person to another person. Therefore, it is known as vector or carrier host. Female Anopheles mosquito is known as carrier host because it carries this parasite. Due to requirement of two hosts for completion of the life cycle, Plasmodium is known as a digenetic parasite. Di means two. Two host means one is man and another is malarial and another is the mosquito, female Anopheles mosquito. So this parasite is known as digenetic parasite. The life cycle of Plasmodium takes place in two steps, such as that is asexual cycle and sexual cycle. The asexual cycle is completed in man and involves in three stages or phases, that is pre-erythrocytic phase, number 2 is exoerythrocytic phase, and number 3 is erythrocytic phase. The asexual cycle is otherwise known as schizogony, and the daughter individual produced at each phase of this cycle are named with the suffix merozoites. The daughter individual of each cycle is known as merozoites. The sexual cycle is otherwise known as sporogony. It is completed in the body of a female Anopheles mosquito. The daughter individuals produced in this cycle are called as sporozoites. The daughter individuals of the asexual cycle are merozoites, whereas the daughter individuals of sexual cycle are known as sporozoites. Then the asexual life cycle. This life cycle starts in men when an infected female Anopheles mosquito bites a healthy person. To suck blood, thousands of sporozoites are injected into the bloodstream of man. The sporozoite is the infective stage of the parasite. Structure of the sporozoite. Each sporozoite is a minute, slender and sickle-shaped in appearance. The sporozoite is a minute, slender-like structure. Its body is pointed at both the ends. Here is the sporozoite. Both the ends, the body is pointed at both the ends. And slightly curved towards one side. The size of the sporozoite is about 14 micron in length and 1 micron in breadth. The body remains covered externally by a thin, elastic and non-resistant cuticle. Externally, the body is covered by cuticle or pellicle. The middle part of the body is thicker due to presence of nucleus. The sporozoite is motile. The sporozoite is motile and shows a gliding movement due to the contraction and relaxation of the microtubule. Here is the structure of sporozoites. It is the electron microscopic structure and it is the compound microscopic structure. This is the apical cap. These are the microtubules of the pellicle. It is the outermost covering, then plasma lemma, then this is the nucleus and this is the mitochondria. Then we have to discuss about the different phases, that is pre-erythrocytic phase. Number one is pre-erythrocytic phase. After the entry of the sporozoite into the blood of human, they enter into the liver cell. Then inside the liver cell, the sporozoite loses its shape and becomes round and non-pigmented. At this stage, it is called as cryptozoite. The cryptozoite is the trophozoite or feeding stage of the parasite. Trophozoite is the feeding stage of the parasite. It feeds and attains a full-grown size known as senescent. The senescent multiplies asexually by multiple fission and develops into cryptomerozoites. Then exoerythrocytic phase. Some of the cryptomerozoites enter into the new liver cell in order to continue their life cycle. Inside the new liver cell, each becomes a trophozoite called metacryptozoite. After full grown, the metacryptozoite becomes a senescent and reproduces by multiple fission known as schizogony. As a result, the thousands of daughter individuals are produced called as metacryptomerozoites. Both the pre-erythrocytic and exoerythrocytic schizogony takes place in liver, so it is otherwise known as liver schizogony. Then the erythrocytic phase. This phase starts when the red blood corpuscles of the human are invaded either by pre-erythrocytic cryptomerozoite or exoerythrocytic micrometacryptomerozoite. Each merozoite inside the RBC becomes a trophozoite and then into a senescent. There are so many changes takes place in a senescent. Then the senescent multiplies and forms erythrocytic merozoite. The erythrocytic cycle in the RBC takes about 48 hours to be completed. Some of the merozoites after entering into the RBC do not develop into senescent but develop into a compact and rounded sexual forms called gametocytes. The gametocytes are of two types, that is male or micro gametocytes and female or macro gametocytes. Both the gametocytes are unable to develop further in the blood of men due to warm-blooded animal. Man is a warm-blooded animal, so the microgamete or macrogametocytes are not developed inside the body of human. Then the sexual life cycle starts in the body of Anopheles mosquito. When a female Anopheles mosquito bites a malarial patient, the gametocytes along with different stages of the erythrocytic phase enter into the alimentary canal of the mosquito with the bloodstream. In the stomach of mosquito, all the stages of the parasite except the gametocytes are digested by the action of digestive juice. In the alimentary canal of the mosquito, the gametocytes are developed into gametes. The microgametocytes are developed into male gametes and the macrogametocytes are developed into the macrogamete or ovum. Then the two gametes fuse with each other to produce a single cell called as zygote. The zygote undergoes encysted condition and is known as oocyst. The oocyst undergoes repeated multiplication inside the body of the mosquito, known as sporogony, and the individuals are called as sporozoite. When the sporozoite reach the salivary glands of the mosquito, then the mosquito becomes infective. When such mosquito bites a healthy person, then a man got infection. Then symptoms of malaria. The symptom is followed by rapid increase in the body temperature up to about 103 degree to 104 degree Fahrenheit. Chillness, headache, muscular pains, nausea and sometimes insomnia occurs. That means nausea means vomiting. Tongue becomes thickly coated, mouth becomes dry and patient loses appetite. The high temperature remains for about 6 to 10 hours. Then it decreases to become normal and profuse sweating takes place. Then diagnosis. The disease can be diagnosed by taking the blood of the patient and examining the blood in the laboratory. Treatment. The treatment of the disease is possible by killing the different stages of the parasite with the help of several anti-malarial drugs. Previously, quinine was used for the purpose, but nowadays several anti-malarial drugs such as chloroquine, plasmocin, chemoquine, pentaquine, etc. are used for the treatment. Then prevention. The prevention of malarial parasite can be achieved by using small doses of quinine daily, taking proper care of the patient, by sleeping under mosquito net and applying mosquito repellent cream or odomos over the body so that the mosquito cannot bite. Then control of the disease. Malaria control. Malaria can be controlled by taking the following measures. Number one is mosquitoes should be killed from human habitations by spraying DDT. Insecticides should be applied in ditches and drains so that the breeding of the mosquitoes can be stopped. Larvivorous fishes like Gambusia and Levistes should be introduced into the ponds and drains. The mosquito net should be used at night while sleeping. NMEP, National Malarial Eradication Program. This program is taking place since past several years. A massive malaria eradication program by the joint collaboration with the WHO and Indian Council of Medical Research (ICMR), with the joint collaboration of WHO, World Health Organization and ICMR. Its main aim is to control and eradication of malaria in our country. In this program, 3 effective methods are employed for successful control, that is, destruction of mosquito, prevention of infection, simultaneous treatment of all patients. These are the 3 methods which help to control the eradication of malarial program. Types of malaria. In human beings, generally 4 types of malaria are seen there. Number 1 is benign tertian malaria. This type of malaria is caused by Plasmodium vivax. In this malaria, fever comes at an interval of 48 hours, every 48 hours. The incubation period is 12 to 14 days. It is widely spread in both tropical and temperate zones. Then, number 2 is mild tertian malaria. It is caused by Plasmodium ovale. Fever comes at an interval of every 48 to 50 hours. The incubation period varies from 11 to 16 days. It is found in West Africa and South America. Then quatern malaria. This type of malaria is caused by Plasmodium malaria. Plasmodium malaria. Fever comes at an interval of every 72 hours. The incubation period varies from 28 to 37 days. It is found in both tropical and temperate zone, but not very widespread. Then malignant tertian malaria. This type of malaria is caused by Plasmodium falciparum. Fever comes irregularly at an interval of every 36 to 48 hours. The death rate is high in this type. In this type of malaria, the death rate is high. So this species is known as killer variety. The incubation period varies from 7 to 27 days. It is most dangerous type and is very common in tropical countries. This type of malaria is also known as pernicious malaria or endemic malaria. This is end of your chapter, malarial fever. Thank you.